Good morning, everybody, and good afternoon to all of you in other places around the world. I'd like to welcome you to our webcast today. My name is Gary Schroth. I'm a distinguished scientist at Illumina. I work in our R&D group in a team we call the Applications Group. And today I'm going to talk about studying the transcriptome with next-generation sequencing. I will say up front, we would really love to have your questions, and you can submit the questions in the question and answer area on the board that is part of the webcast. So if you can just write out the questions, we'll go through them at the end of the meeting, at the end of the webcast, as many as we can. So I'll just jump right in with the presentation. Today I'm going to be talking about the transcriptome and how you can use next-generation sequencing to study the transcriptome. The transcriptome, as we know, is very complex. RNA has many, many functions and functionalities inside the cell. It's not only complex, it's also very dynamic. RNA levels go up and down depending upon what type of cell it is and what the environment of the cell is in. And so this makes it quite challenging to study the transcriptome, but also quite interesting. So the other interesting thing about RNA is that there are many types of subpopulations of RNA in eukaryotic cells, from coding RNAs, messenger RNA, and even those have very highly expressed versions, over to non-coding RNAs, which have been just a boom in the last 10 years, the study of non-coding RNAs, from small RNAs, microRNAs, siRNAs, to other transcriptional RNAs that are involved in the transcriptional process, and then, of course, link RNAs. One of the interesting things is if you just look in general, most coding RNAs are polyadenylated, so the fully processed transcripts have a poly-A tail, whereas I wouldn't say most, but many of the non-coding RNAs are not polyadenylated. MicroRNAs, of course, are not polyadenylated. Many of these regulators are not polyadenylated. Link RNAs are kind of an interesting class. Some are polyadenylated and some are not. But the point is if you do a poly-A preparation, as I'm going to talk about, you tend to capture just a subset of all of these molecules. The other thing about RNA that makes it interesting is that, unlike DNA, which is fairly monotonous structure, RNA comes in all kinds of sizes and shapes, and stabilities is really important. In general, RNA is much more unstable and fragile than DNA. But probably the most important thing about RNA that makes it an interesting thing to study with next-gen sequencing is the copy number variation. And so if you look at the dynamic range of transcription in a eukaryotic cell, it's truly enormous. It's over 3, 4, sometimes even 5 logs of dynamic range if you consider the most highly expressed transcripts that we can detect down to the lowest expressed transcripts that we can detect. And this makes it interesting when you're working with next-gen sequencing, which is essentially a counting application. RNA-Seq is essentially a counting application, and you essentially are measuring by sequencing the number of molecules you have associated with each of these transcripts. And so what we find is that transcripts up at this end of the spectrum are very easy to study. In fact, we get plenty of data, plenty of reads associated with transcripts up here. And transcripts down at this end are more challenging to study. And frankly, all the discussion about the sensitivity of RNA-Seq versus any other method, or from run to run, or how many reads do you need, all that debate always centers on genes down at this end of the spectrum. From about here on up, you only need 5 or 10 million reads to actually see everything you want to see in these genes. It's really about what goes on at this low end. So we'll have some recommendations throughout this webcast as to what we would recommend for the various preps to study some of these genes down at this end. The other really cool thing about RNA-Seq and transcriptomics in general is that it's so dynamic. This is one of my favorite figures. It was actually put together by Brent Gravely of the ModEncode project. It just shows a very simple, relatively simple 24-hour time progression of Drosophila development. And we're looking at just one small region of one chromosome in Drosophila. And you can see the data, the reads on the top strand come from one gene, the reads on the bottom strand come from a gene transcribing in the opposite direction. But you get to see the complexity that's happening just in this one region with this handful of genes throughout development in Drosophila. And essentially you can picture this happening across the genome in any eukaryotic system as you perturb it. And that's what is really amazing about the sort of resolution and insight you can get from RNA-Seq, is that you can really start to look at these dynamic expression changes in amazing detail. And another important concept in transcriptomics is strandedness, as sort of shown in that last figure. You wouldn't be able to see the genes sort of an up and down configuration here, the Watson or the Crick strand, without what we call a stranded prep. And this is just, I've color-coded them here, where this is two standard samples we use, UHR and brain, where I've color-coded the preps red for the top strand and blue for the bottom strand. And you can see that this gene comes in this direction, the transcript from this gene goes in this direction, the transcript from this gene goes in the opposite direction. And so they align data in our prep, because we preserve the strandedness of the data using the TruSeq RNA prep process. We know that this data comes from the top or the bottom strand. And if you look at another region where here's two genes that physically overlap each other, but remember these transcripts are coming from the opposite strands of the DNA, it's very clear in a non-stranded prep, or if you don't consider the strandedness even in your data, you just look at the pile of the reads, you really wouldn't know for these reads in this area, are they assigned to this transcript or that transcript? But if you actually use stranded data, it's very obvious that most of the reads from this pile, in fact, come from the lower transcript and not the upper one. And you can assign reads explicitly to the correct transcript with stranded information. Of course, genes are in eukaryotes are the relationship between the primary transcript and the mature messenger RNA involves splicing and removal of introns. And so that is one of the things that's quite obvious when you look at RNA-Seq data, is you can see that a lot of the genes are spread out across large swaths of the chromosome, creating relatively huge primary transcripts, oftentimes 10, 50, even 100 kb long, that are then spliced into into mature messenger RNA molecules that can be alternatively spliced, as is shown here, but also they're generally much smaller and contain a poly-A tail. So that when you look at different library prep options, you have the choice of doing a poly-A selection where you capture essentially mostly intact mature messenger RNA, or we have a method that we call total RNA-Seq, where you capture everything. In total RNA-Seq, we only remove the ribosomal RNA and essentially sequence everything else in the cell, including a lot of these primary transcripts that you don't see when you do messenger RNA sequencing. So this is just another form of complexity that exists in the eukaryotic transcriptome. The human genome only has 20,000 genes, basically, 20 to 22,000 genes, but it's believed that there are upwards of 500,000 different types of isoforms of transcripts that can be derived from those genes, and that's due to alternative splicing, which can be seen explicitly in RNA-Seq and can be counted in terms of the isoforms in RNA-Seq. And so here's a well-known example of a tissue-specific pattern of expression between these two exons here. You can see these two tissues utilize that exon. This tissue utilizes that exon to a much less degree than it utilizes the exon to the left here. And so this is a very clear example of how you can quantify different splice levels using RNA-Seq. This is just a diagram to show the types of complexity that are observed in alternative splicing. It really can get quite complicated, considering every gene has, on average, 10 to 15 exons, and then you can shuffle those around and remove some, extend the 5' or 3' end, use different splice sites, alternative promoters or different polyadenylation sites. So the complexity of the types of transcripts you can get off of the basic set of 20,000 human genes is quite huge. And that's where very deep RNA sequencing can really help elucidate all of that structure. here at Illumina in my group, we've been working on RNA-Seq for about 7.5 years now, and during that time, the whole process has evolved a lot, and so I just kind of want to highlight some of the things that we've learned over that time. We've worked on many new methods. I mentioned stranded. We've had both stranded and non-stranded methods, and we have many, many varieties of the stranded TruSeq RNA prep kits available today. We have many new methods in terms of comparing poly-A to total RNA kits, as I alluded to a few slides ago. Total RNA has a lot of advantages in that it allows us to look at these non-coding RNAs, many that don't have poly-A tails, and they have been providing a lot of interesting biology to researchers over the last 5 years. And the other thing that's a subtlety in working with the total RNA is that it's much less sensitive to RNA degradation than a poly-A prep. We've also been able to push to lower and even lower input levels. So our first RNA-Seq kit that was released probably 5 years ago required 10 micrograms of total RNA. All of the kits in our sort of suite of kits that we have today require only 100 nanograms of total RNA, and some can even go lower than that. So that makes it much more manageable in terms of the size of the sample, the number of cells that you need to do RNA-Seq. We've developed much more sophisticated methods for looking at degraded RNA, and that's what I'm going to spend a lot of time on during this presentation, is talking about some of our latest methods for getting really good performance from FFPE samples. We've developed single-cell RNA sequencing methods, and these are now widely used around the world, and this is sort of definitely an interesting and growing area. Unfortunately, we won't have a lot of time to talk about that today, but there's a lot of resources out there about single-cell RNA sequencing. And then also just want to highlight that pretty much all of the methods that I'm going to talk about today and the established kits that we have, we collaborate with pretty much all of the vendors who automate protocols, Hamilton, Beckman, TCAN, etc. And many of them have explicitly developed automation protocols for the TruSeq kits. And we actually have a spot on our website where we highlight all the different vendors and which specific kits have been validated by Illumina in collaboration with these vendors for automation. So this next slide gives an overview of the different types of, I would say, sample prep paradigms that there are out there in our kits, and frankly in any method that you're going to use to look at the transcriptome. The first thing you're going to do, as I've mentioned already, is decide if you're going to do a poly-A prep or a total RNA prep. A poly-A messenger RNA prep, the first thing you would do is, in our protocol, is to capture the RNA away from the mostly ribosomal, but also intronic and all the non-coding RNAs and small RNAs. You're going to capture only mature messages and then fragment them, turn them into cDNA, add adapters, and essentially create a library that's appropriate for sequencing. In total RNA, it's a very similar workflow. You start with very similar amounts of material, but instead of capturing the poly-A tail, you remove the ribosomal RNA using a capture-like approach, and then you sequence, create a library out of essentially everything else left behind. There is some sizing that goes on in this, so we don't see microRNAs in this. So when I say everything, it does not include small RNAs. But everything that's about 100 to 150 nucleotides or longer is captured in this process in the cell. And so these are the two major paradigms that all of our kits and most of the sample prep methods that have been published are based upon. What I want to talk about today is actually a new paradigm where it addresses a couple of different problems. One is if the RNA is fragmented, then the poly-A capture can be quite problematic. And instead of capturing full-length RNA, you could imagine that if this RNA were fragmented by a degradation process, or like you get from FFPE samples, that you would capture essentially just the 3' end of this message as opposed to the full-length message. We also, as I've alluded to, total RNA works quite well on degraded samples, so it's relatively independent of degradation, but sometimes, as I'll highlight with FFPE, it requires much deeper sequencing to get a good answer out. And so to address both of these issues, we developed this method that we call TruSeq RNA access. And the access process is actually more similar to an exon capture or exome sequencing process that you might be familiar with in a DNA world than anything we've ever done in the RNA world before. So what we do is we skip both mRNA capture and ribosomal removal. We take all of the total RNA and convert it into a library. So we fragment all of it. We do first and second strand synthesis, add adapters, and essentially at this point we have a library that's ready for sequencing. If we sequenced it, it would almost all of it would be ribosomal RNA because we did not remove the ribosomal RNA. But instead what we do is we use specific probes to capture the coding regions of the messenger RNA that we'd like to sequence, and then we sequence that. So this is a much more focused sort of RNA-Seq experiment than even the mRNA-Seq that we've done in the past. The other interesting thing about this is because we use all the RNA, we don't separate out mRNA or removal of the ribosomal up front. We can actually start with about 10 times less material for this. So this makes this assay even more sensitive than our other two assays in terms of input levels. When you look at what you get from these three preps in a little bit more detail, so this is running the prep, these three varieties of the prep, let's call it the RNA access, mRNA, and total RNA. So these are three of our kits that essentially have the same basic chemistry behind them, but it's either capturing poly A tail, removing ribosomal first, or capturing just the coding regions. And this is on the same sample, MCF7. And you can see you get fairly significantly different results depending upon what type of prep you use. And I think this is something that you need to be cognizant of when you're thinking about doing a transcriptome experiment. What type of sample do you have? What types of things would you like to capture? What types of things are you interested in looking at in the data? Because you get significantly different views of the transcriptome. These are all perfectly legitimate. They're all perfectly correct, if you will. It's not like one is more biased than the other. They're just different slices of the same pie. So with the total RNA, you get a lot more intronic and non-coding RNAs showing up, intergenic in this pile, whereas with RNA access, we get almost exclusively, close to 80% of the reads actually are hitting coding regions, which is where we put most of our probes. So this is what the data looks like if you look at it on a genome browser, and you'll see throughout this presentation that I really like looking at the data on the genome browser. I think it's really important to sort of validate with your eyes across the genome browser how the data is behaving. And you can see here is internal control gene like GAPDH, fairly highly expressed. You can see the pattern of signal we get in an mRNA prep and a total RNA prep. And then this is the capture prep that we're working with in this called RNA access. And actually down here in green are the individual probes or the regions that we were capturing in this. So you can see that we do a pretty good job of capturing the RNA from end to end, and we get very robust numbers that correlate quite well with the numbers that we would get from either mRNA or total RNA, and I'll show you that as well. This is also another example of comparing the three preps. This is a little bit more complicated area of the genome where we actually have a gene overlapping here. I'm not showing actually the strandedness of this. This data is stranded. I'll show some examples of that later. But I wanted to just highlight, here's where the regions that we're capturing, and you can see that we don't, for instance, capture this intronic signal at all with our probe set in RNA access, which is by design. Instead, we just capture the coding regions and then, of course, the linkages between them, as I'll get to in a second. Here's probably one of the more interesting examples of a protein coding gene that is not polyadenylated. All of the histone genes, or most all of the histone genes, are fairly highly expressed. They obviously code for structural proteins, the histones, but they are notoriously not polyadenylated. So in all of our preps over the years, whether we're doing microarrays or RNA-Seq, if you're doing a poly-A-based approach, essentially the histone genes are completely lost. Whereas they show up quite well in total RNA, and they show up in the RNA access process because we put a probe there in the middle of that short gene. And so that's an interesting case of a protein coding region that is, or protein coding transcript, mRNA, that is not polyadenylated. The other thing that I'd like to talk about is sort of what we have developed over, working with RNA-Seq for the last, as I said, 7 going on 8 years now, is we've developed a sort of a system for RNA-Seq analysis that involves the library preparation process and all the variants we have, the sequencing across any of our platforms, and then more recently, in the last year or so, we've been working on more sophisticated analysis tools that our customers can use to analyze their data coming off the Illumina systems. And so when we talk about the RNA-Seq system that we use in my group, we are talking about using one of these 3 types of TruSeq RNA prep kits, the mRNA, total RNA, or RNA access. And our standard for sequencing is paired 75 base pair reads. We have different recommendations of read depths depending upon the sample prep or the sample type and the kit used, and I'll have a table later on that will give sort of some rough numbers for that. And then the analysis using our TopHat cufflink software that is provided in the BaseSpace environment. So essentially, all of our sequencers are designed so that they can stream directly to BaseSpace, which is a cloud-based environment, or you can have something called BaseSpace on-site, which is a computer on-site that mimics what happens in the cloud-based environment. Either way, you could run these apps, and I'm going to show you some output from these apps. My group uses them on every run that we do, and we find it's really important to get all of the sort of important QC metrics that we run as a baseline analysis for sure every time we do an RNA-Seq experiment. This is just to remind me to tell you that actually it's not just RNA-Seq apps that we have in BaseSpace. We have apps for sort of the top common biological workflows, and many more are up there in the cloud at the moment. But we earlier this year we launched sort of 4 major new apps for exome, RNA-Seq, whole genome sequencing, and tumor normal comparison of genome or exome sequencing. These are fully available. You just go to BaseSpace, sign up for an account, and actually you can test them out for free now and analyze even your own data. This is a little bit more detail about the RNA-Seq app. It's actually 2 apps. The first one is a TopHat alignment app. The second is Cufflink's assembly and differential expression app. We tried to make these extremely easy to use, and I like this quote from one of our beta testers from several months ago, who basically, this is exactly the kind of thing we're going for. He says, working with next-gen sequencing data as easy as launching an app in my iPhone. It really is that easy to use. You really just load your data into the process. There's a very few options that you have to choose. You press go, and then you'll get an email saying that the analysis is done in a few hours, and then you can start looking through your results. This is the type of report we get out for the TopHat. Like I said, it has a lot of the key metrics. So this is essentially the same TopHat that many, probably 85% of the people in the world who do RNA-Seq use TopHat in some way. We've incorporated into a pipeline that then has an output and sort of a report that we've worked on that we think is useful for QCing the data. Like, for instance, things like insert sizes across the samples, where the data maps. So after it's mapped, does it map to coding regions, UTRs, introns, or intergenic? And you can see right away, this is a dramatic difference between using an mRNA-Seq prep on the top or a total RNA-Seq prep on this sample, and then kind of replicates on a different sample below. As sort of shown in that bar graph a few slides ago. And then also coverage, 5' to 3' bias or coverage differences. This will change a lot if you're doing a poly A prep and you have some degradation in your sample. You will, of course, get more of a 3' bias. And so that leads me to the next part of my talk. I do want to talk about degraded samples, and in particular FFPE samples, which are probably some of the more notoriously difficult samples to work with any kind of expression technology, including RNA-Seq. But I think we've developed some really nice methods that work quite well with a lot of these samples. And so I want to kind of go through that with you. for the next few minutes. But just to remind you, part of the challenge with working with FFPE samples is that, and I guess I should remind people what I mean by FFPE. So FFPE stands for formalin-fixed paraffin-bedded samples. And in general, when a surgeon removes a tissue from a patient, this is the way it is preserved for pathologists to look at the sample, is it's first fixed with formalin for a couple of hours, and then that fixed material is then embedded in a paraffin block so that it can be sliced and looked at under a microscope and stained with different histological stains. And also, you have the ability to purify RNA out of those blocks and study them with RNA-Seq. The problem is that after all that processing, the RNA can sometimes be quite degraded, depending upon the tissue and the time and how long it's sat at room temperature and all kinds of variables. But the bottom line is, this is what pristine quality RNA will look like, and this is what it would look like oftentimes from an FFPE block. And so what my group has done is we have sort of a standard set of tumor normal pairs, lung, stomach, colon, and breast tissues, that we have this nice tumor normal pair set. And what I like about this is it kind of has a wide breadth of quality. From this breast tumor sample has actually not that horribly degraded, whereas this stomach normal actually is so degraded I sometimes refer to this sample as dust. And in fact, just to be upfront, this is probably too degraded for us to analyze with RNA-Seq or frankly with any other technology that's out there. It's just, there's no fragments in there that are much above 100 bases long. So another more subtle challenge and that goes on in FFPE samples, and we've been analyzing FFPE samples with RNA-Seq for 3 or 4 years now, and like a lot of people who have been doing that, what we find is this unusual artifact that coming from the FFPE that no one, as I know, has a way around this, is that if you look at the same tissue, and let's say you have a fresh version of that tissue and an FFPE version of the tissue, and you do a poly-A selected mRNA-Seq on the fresh, you'll see a nice pattern where you get spikes right on the exons, as you would expect. If you look at the same data from total RNA, you'll get a very similar looking pattern with perhaps more intronic and intergenic signal. If you take that same sample that has then been fixed and do any of our FFPE preps, the older preps, like say Ribo0 or total RNA type prep, you get a similar pattern, but what you'll notice is that the exons are all sort of diminished in size, and I refer to this as exonic loss. And this is something that we just have seen over and over and over in FFPE studies. And so that's where the RNA access process comes into play to help us overcome this exonic loss. It sort of hit us a couple years ago that if we used exon capture or exome capture like we do in our DNA world, we should be able to essentially recover the yield of the data that we want, which is on these exons. And so we developed this process where we start with total RNA. We, like I alluded to in one of the previous slides, we make a stranded RNA-Seq library. We take about 200 nanograms of that library and pool 4 libraries together, so 4-plex, total of 800 nanograms of library. And then we do an exome capture, and then we sequence that. And so essentially this is sort of like blending together 2 of our other kits that we've sold historically. The TruSeq stranded RNA kit, which, as I've said, we have several different flavors of that. So it has the same basic cDNA conversion strategy in there. And then the rapid capture exome type of capture and wash that we use in our exome sequencing. But in fact, it turns out that doing this for RNA is even simpler. So we were able to streamline the process for the hybridizations and wash with the rapid capture and turn it from a 2.5-day process into a 1-day process for RNA. The RNA library is much less complex than a genomic library, so it turns out to be quite easy to capture the exome out of a library from RNA. So this is what the data looks like, and this shows you that advantage of using capture in FFP. All 3 of these browser shots are showing the same gene. It's a really interesting cancer gene called FGFR2, And this is using our sort of the state-of-the-art method as of 6 months ago for looking at FFPE, which would be using our total TruSeq stranded total RNA prep kit. And the nice thing is you can get full-length coverage of this and all the other transcripts, but you need quite a lot of reads to get good coverage on the exons. So this track is showing, and these are all scaled to the same scale, what the data coming from this transcript in this region of the genome looks like with 200 million reads derived from a high-seq run. So that's essentially almost one full sample per lane of high-seq versus, and this shows what it would look like if you sort of down-sampled that down to 30 million reads, versus 25 million reads using this RNA access process where we capture the exons only. And you see, not only does it clean up the pattern and you don't actually detect any of these intronic reads anymore, it gives you much better signals to noise on the things we are interested in, which is the exons and the junctions between the exons. And so before I get any more into the data, I do want to spend a little bit of time talking about another important thing that we have developed during the development of the RNA access kit, and that is a better QC metric for looking at FFPE, RNA samples from FFPE. And so if you, I wish I would have highlighted that, but if you look at the RIN number for all of these samples, they would all be basically 2 point something. The RIN number is called the RNA integrity number that comes from the Agilent bioanalyzer, sort of reported out automatically. It's really designed to look on, to calculate RNA that is less degraded than this. So more closer to intact RNA than these RNAs. When you get down into this regime, the RIN number is really not appropriate. And so what we found is that we did something much more, I think, intuitively obvious, is if you just draw a line here at 200 nucleotides for each of these traces, and you just calculate the amount of material that's more than 200 nucleotides long, meaning we would have high confidence that that would make a library and be sequenceable, you can see that there's a nice trend here between this breast tumor sample and, as I mentioned, this stomach tumor sample in terms of the amount of material that is essentially available to create a library. And so obviously this sample is much more degraded. This DV200 number is much lower. And in fact, what we find is a very nice and reproducible standard curve that we have been able to replicate this across many studies now, that this number, more so than anything else we've ever looked at in terms of RNA QC, allows us to predict the output of the library that we're going to get by going through a sort of standardized preparation process. And this is important because we need to generate a certain amount of material to then use in the capture process. So, and in fact, we've gotten, some might say, even overly sophisticated about this in terms of we can now classify samples into very good ones where we'd be very confident that 20 nanograms of FFP material will work well, to poor samples where I might go up to even 100 nanograms, to samples that we can, and this is another key feature of this QC, is that oftentimes we can tell before we even start the library prep process or the sequencing process, which are obviously both somewhat expensive. You don't want to do that on samples where you have no hope of getting results. And so this allows us to essentially pre-qualify samples for the RNA access prep process. And what we find is that in the real world, this is not that limiting. We find very few studies where most of the samples are below this threshold. In fact, it's usually the opposite. So here's one of the poor quality samples. Like I said, this would be not recommended for, this is the stomach normal. The DV200 is 8%. We've tried hard to make a library out of this. We can make a teeny little bit of library, but it's really not a robust process. And so, you know, this RNA is probably degraded because normal stomach tissue has a lot of RNase activity in it, and so the RNA is probably very difficult to get out of stomach tissue. And so, like I said, the nice thing about this is that it turns out to be very predictive. And if you look at either yield versus sort of DV200 number, also I put on here the RINs just to show you that they don't correlate at all with this sort of output. You can see that with these very degraded samples, we can make enough library for sequencing. We really would like to hit this 200 nanogram threshold. So you can see that With the poorest quality samples, if we go up to 80 nanograms of input, we can get above that 200 nanogram output quite readily, and that allows us to then move forward into the enrichment. So that's sort of the goal of this part of the sample prep process. So, like I said, we generate 200 nanograms of library, RNA-Seq library. We then use that in the exome capture process and are ready for sequencing. So the nice thing about this process, like frankly all of our kits, we make very reproducible and quantitative libraries out of this. And even though this is a capture process, what has sort of surprised us about this entire thing is that how quantitative this is. We capture across the full dynamic range. We have never seen any evidence of sort of saturation of probes at this end or not being able to capture things at this end. In a lot of ways, this looks very much like standard RNA-Seq data. And so we can see gene expression across 5 orders of magnitude like we would expect to see. And once again, the whole point of this is that it focuses the sequencing on the regions that you're sort of counting anyway. This is just a zoom in on that region, just to show you how dramatic this is across this particular gene. And this is one of the sort of worst offenders, I would say. It's got a lot of intronic signal and a lot of that. You just see how well that's cleaned up by the capture process. Of course, we know exome enrichment works very well. You know, there literally have been tens of thousands of human exomes been sequenced with these similar kinds of reagents, but applying it to a DNA library prep against the entire genome. Here we're applying it to an RNA library prep against essentially the transcriptome, and it's even cleaner in a lot of ways. So this is the table I was alluding to earlier. This is where we get the added benefit of the RNA access process. So I've already shown you how you could actually use less material, 10, certainly 20 nanograms for high quality FFPE samples as opposed to 100 or more. So, you know, 5 to 10-fold reduction in input into the process, but then you can also get a much more bang for your buck, so to speak, if you're doing RNA access on FFPE in terms of the sequencing costs as well. So this is just showing no matter what coverage you're shooting for, you know, and this is just kind of average coverage across the genome or the transcriptome, and this is read depth. What this shows is that, let's say you're used to working with fresh, high-quality samples and collecting 25 million reads from one of our systems. What we would recommend that if you're using that sample and you're doing total RNA, we would recommend doubling your output to get the same sort of coverage, just because so many of the reads go to the intronic signals or non-coding RNAs. With RNA access, frankly, 25 million reads is more, gives you better coverage, but we're just, for the sake of this discussion, we're going to make these sort of equivalent. They're fairly close. But the point is that if you have degraded samples, and especially fixed degraded samples like FFPE, first off, we would never recommend doing an mRNA prep on those. But if you're doing a total RNA prep like you would have done in the past few years, you sometimes have to go, as an example I was showing you, 200 million reads to get good signal. And this is the cool thing about the RNA access process. Because we essentially are capturing the same subset of RNAs out of the pool, whether it's fresh, high quality, fresh degraded, or even FFPE, 25 million reads looks very, very similar in terms of the coverage of the genome. And so what this means in practical terms is that if you're studying FFPE, instead of running 1 sample per lane of Hi-Seq, you can now run 8 samples per lane of Hi-Seq and get actually, frankly, in a lot of ways, much better looking data. This is actually some very interesting data that was generated by Chuck Peru's lab at University of North Carolina. They actually were testing out the RNA access kit several months ago and did this very nice comparison, comparing total RNA-Seq to RNA access in fresh samples, total RNA-Seq to RNA access in FFPE, and then access only in FFPE versus fresh. And this is absolute count. So this is the absolute sort of FPKM, RPKM signals compared to one another. And you can see these correlate quite well. Even in highly degraded samples versus fresh, the RNA access kit's highly correlated. So this confirms what I was saying a few minutes ago, that they essentially, even though these 3 processes are very different And in fact, this enrichment process looks like it has the opportunity potentially to bias either for large stuff or small stuff. Actually, we see no evidence of that. We recapitulate the dynamic range, and in fact, the quantitative dynamic range of our other two standard kits, even sort of taking into account the fixation process versus fresh. So this is really quite impressive. So once again, this is another one of those bar plots to show you the dramatic difference between using total RNA-Seq on FFPE versus the RNA access process. This is, like I said, the output of our BaseSpace app. Once again, it sort of simply bins the data after mapping to coding regions, UTRs, introns, or intergenic. And you can see that how the RNA access, just as those vivid images I've been showing you from the FGFR gene, it's doing that across the whole transcriptome, in that pretty much all of the data comes from coding regions and UTRs, very little from intergenic or intronic. And so essentially we get what we probe for in most of in almost all of the data. And so I showed you how the absolute counts correlate quite well. So really this shouldn't be much of a surprise, but what we find is that when we look at the ratio counts, or the fold change ratios that we would get from this new process, the capture process, RNA access, versus the total RNA process, which I would still refer to the total RNA process as probably the most unbiased way to look at the transcriptome. Once you remove the ribosomal RNA and sequence everything else, that really is the most unbiased way to look at all of your transcripts. And you know, frankly, I have to admit, when we were developing the RNA access kit, I expected there to be some bias due to the fact that we were going for the capture and enrichment. And in fact, as Chuck's data showed a couple of slides ago, and this data shows, it's remarkably well correlated with the data that we get from the completely unbiased total RNA prep or the enriched RNA access prep. If you just look at the fold change between these two tissues in a fresh RNA sample, or even between a tumor normal pair and an FFPE RNA sample, that is as good a correlation as we get between any two RNA-Seq technologies, or frankly any two types of gene expression technologies. And so once again, we really have seen zero evidence or data to support the fact that the enrichment process is in any way biasing the samples. Of course, it does bias it in terms of you get what you probe for, but it doesn't bias it in terms of the quantitative nature of what it's capturing. The other thing I wanted to just remind you of is that this data, even though we target, you can see where the probes are down here, we're targeting exonic regions, frankly the same regions that we target in the genomic DNA. Because that we're not probing genomic DNA, we're probing this as a transcript, a cDNA molecule. We pull out all of the junctions, even though there is not any explicit probe in here to a junction in the RNA. We capture the junctions quite efficiently because we effectively capture this exon and that exon, so we pull down fragments that also contain the junctions. And this is just to remind me to tell you that this data is stranded. It uses our stranded RNA-Seq process, so you can see that the data piles up on this strand for this gene or this strand for this other gene. And the junctions that you get out, these are quantitative piles of junctions or reads that map to splice junctions, is also quantitative and also stranded. This just shows some statistics to support that. If you look at either fresh samples or FFPE samples, with the RNA access kit, just as I've alluded to before, we get very sort of similar results from even really different samples, and this bears true here. Basically, all of these are in the very similar 35 to 40% of the reads are hitting a splice junction. But you can see in total RNA, as we've already discussed, you can get down to actually quite a small amount of the data is hitting exons and thus splice junctions in a total RNA prep, which is, you know, this is back to, once again, the sort of 8-fold difference in coverage that you need to get similar coverage. So the reason I'm talking about splice junctions is also, you know, to point out that you can use the data for alternative splicing, but also to lead into the next topic, which is fusion transcript detection. Obviously, many of the studies where we have these FFPE samples are derived from cancer, and so the point is that can we use this as cancer researchers to, even though it's an entirely targeted approach, can we also still, quote unquote, discover new fusion transcripts? And so it turns out this works remarkably well for that as well. So here is some raw data and the output of TopHat fusion. I'm highlighting here the numbers of reads that we get that span the famous BCR-ABL fusion that you can see in our standard RNA UHRR. And I'm comparing the RNA access process with sort of 18 to 30 million reads versus the ribo-zero or total RNA-Seq process in the 50 to 100 million reads. You can see that actually we get better coverage of this particular junction, fusion junction, using the RNA access even compared to the sort of unbiased total RNA-Seq. And this is once again due to the sort of focused enrichment on the exome. Now, the one thing I want to highlight is that, as I said before, there is not a single probe in this probe set that explicitly targets BCR-ABL fusion transcripts, but we effectively capture the BCR junction on one side and the ABL junction on the other side, and when we capture those, we pull down the fusion as well. And so this is sort of a blow-up of that region, showing some of the reads that either touch the junction or are gray if they're on one side or the other. These are paired-end reads. And you can see here's the actual probe that we have for the BCR side and the ABL side, the enrichment probe. And we're basically pulling down fragments that span this region. And when we map them back, it's very clear that we are detecting the BCR-ABL fusion. Here's another one that we detect quite readily. It's GAS6 RASA3 fusion. It's also known to be in UHR. And over time, we've done this now for lots of different fusions, and I can say that so far we haven't seemed to miss any fusions that we knew were in a sample using this process. We also have been able to use this to discover fusions, and here's a small snippet of data that we generated in collaboration with Boris Bastian lab at UCSF, very interested in melanoma, and we used, these are sort of real cancer melanoma patient samples. We started with 20 nanograms of input RNA. These plots show the quality of the RNA. They were above our 30% cutoff of DV200. And so, but you can see they're legitimately and highly degraded. We collected about, in this experiment, 14 to 30 million reads. And what you could see is that we were able to detect a couple of interesting NTRK fusions and a BRAF fusion in this particular patient sample. Frankly, this group was sort of looking for that in these. They consider it sort of a positive control, but still, I think we're all surprised by the read depth and the amount of data that we got from a relatively small amount of RNA-seq data. And of course, also bearing in mind that the initial starting material looked as very poor quality. And so I think this is this shows that even though we have no fusions or no probes in this data set against any of these fusions, we can still use this highly targeted enrichment set to discover novel fusions. And so I will remind you that our exome capture probe set is enormous. It targets over 21,000 genes. It has almost 500,000 probes in it. So there are very few exons that we miss that might be involved in a fusion. And as I said, we haven't found one yet. So the other nice thing about this, we are using just a standard TopHat fusion algorithm. There's lots of algorithms out there in the world that claim to be better or different, and of course you're free to use that data or those algorithms on this data. I will just say that in the TopHat, it's as easy as clicking that box when you set off the run. That's one of the few options that you have, actually, and then you can essentially detect fusions and generate those numbers like I've been showing you throughout this presentation. So, in summary, this RNA access is a robust new, essentially sample prep paradigm that sort of sits alongside our poly-A preps and our total RNA preps. And now we're developing a whole line of products based upon this sort of enrichment method. And so the first kit that I talked about is the entire, sort of whole transcriptome enrichment. Allows us to look at essentially all known REF-seq genes and look at very quantitative differential gene expression. I showed you how we can use samples down to 10 nanogram inputs for fresh, even some FFPE, certainly down to 20 nanograms for most FFPEs. And then you can look at quantitative accuracy of differential expression. All three of these preps compare to each other relatively well in terms of both absolute counts, but also especially fold change differences. And then we can use this to get enhanced sensitivities for splice junctions and fusions. And so, and also this is a very efficient, effective way for fusion discovery in cancer samples, even without any a priori knowledge of which fusions you're going for. And so this is just to remind you that this is a part of our suite of sample prep solutions for RNA library prep. I talked a lot about the stranded total RNA kits. We have those for plants, for a globin kit for blood, for pulling out, removing mitochondrial RNA as well, that we call ribozero gold, coupled with ribozero gold. We, of course, have an mRNA kit and the TruSeq RNA access. And I didn't have time to mention these much today, but we also have a microRNA kit that is called the TruSeq small RNA kit. And then we have a very targeted kit that does panels of gene expression very efficiently and very inexpensively to look at gene expression panels. We call it TruSeq targeted RNA expression. This kit is fully available, the one I've been talking about mainly through this presentation, the RNA Access Library Prep Kit. There's more information on our website about this, obviously. And essentially, this is part of our growing and we hope complete portfolio that we're developing, where we're helping people start with sample, get good data, and essentially have as easy a workflow as possible, sort of organized around this BaseSpace sort of set of tools, where we have library prep solutions, sequencing solutions, and downstream analysis solutions, so that this entire process can be as easy as it possibly can in terms of starting with total RNAs and getting results like I've been showing you throughout this presentation. So with that, I would like to thank everybody for attending this webcast and for your attention. I'd be interested in getting feedback from you guys. This is my email and my Twitter handle. And with that, I would going to look over here in the Q&A panel and see if what kind of questions we've gotten. One of the first questions here, I think, is actually a really important question, which is the question is, why is total RNA less prone to degradation than poly-A RNA? And that's a really important concept. So if you think back to what we did in the poly-A capture process, the first thing we do is take the RNA with a bead with a poly-T primer and essentially pull out the poly-A linked messenger RNA. If the RNA is degraded, what you pull out in that step is really just the 3' stub of the RNA, the original RNA molecule. So you can imagine if your RNA is degraded to an average size of 200, 300 bases long, then when you do a poly-A capture, you're going to pull out roughly 200, 300 base pair end pieces of every transcript. And so that causes a lot of what we call 3' bias. In the total RNA prep, because it is not based on poly-A at all, we essentially are sequencing all the RNA. The only thing we're removing from the pot is ribosomal RNA. And our removal process that we use, this ribozero technology, is quite effective whether the RNA is degraded or not. So we remove all the little bits of ribosomal RNA, and then what's left behind is the full representation from end to end. So when we randomly prime that and convert that into cDNA, we really see very little evidence of 3' bias like you would in the poly-A probe. Let's see. Let's see what I'm trying to look through the protocol or the differences here. There is a question about, can we use this kit for looking at bacterial populations or mixed samples, eukaryotic and prokaryotic? So you could. We don't have probes in there for the prokaryotic transcriptomes yet. That's something that we're considering in the future, is to have essentially more the ability to create custom sets. That's not ready for immediate release or anything, but I think that you could potentially use this same approach to pull out any RNA out of the population. You could also use this same kit if you had a mixture of eukaryotic and prokaryotic samples to study the eukaryotic part. The human part would come out just basically the same way as it does from an FFPE block. But in terms of seeing both eukaryotic and prokaryotic samples, we would need to create a different probe set. The other question is, could you comment on the significant differences between Illumina's RNA-Seq and targeted RNA expression analysis pipelines and BaseSpace apps? So just as we have a complete set of RNA-Seq sample prep solutions, we also have a developing set of apps as well. And so we have the TopHat cufflinks app that I've already mentioned. We also have a relatively new one called RNA Express, which uses a slightly different alignment and counting algorithm. It's based upon the star aligner as opposed to the TopHat aligner, and it's based upon what is called DE-Seq for differential expression counting analysis, which is a little bit different than cufflinks. So both of those options are actually available. It's actually kind of interesting to run the pipeline on the same data set, these two different pipelines, and see what sort of small differences you detect, because sometimes those are important and sometimes they are the result of different apps. I will say that that the RNA access data, even though it is enriched in terms of we are pulling out the coding region, all the apps work very well, and in fact, to the apps, this data seems to look like very high-quality mRNA-Seq data, because you really are just getting the coding regions of the messages. And so there's some things that are a little bit different about the apps. The coverage differences are kind of characteristic between RNA access and mRNA or total RNA-Seq. But in general, the apps don't recognize any real difference between these, and they can all be used with these processes. So this is a sample. This is kind of a complicated question. The question is, we've seen that partially degraded RNA samples bias gene representation compared to pristine samples, RIN of 7 to 8 versus 9.5. The statistical power to identify differentially expressed genes suffers, so we have excluded these samples. Is the access or RNA depletion method more resistant to this bias? And we do not see these biases when comparing treated versus control FFPE with varying average sizes. So this is a complicated discussion, but basically, a lot of core labs, if I understand this question correctly, a lot of core labs will say we won't touch your samples unless the RIN is 7 or 8 or higher. But even if it is 7 and you're comparing it to some of your samples have a RIN of 9 or 9.5, which as this question says, I would call those pristine samples. If you have half of your set is pristine samples and half are not, and you're using a mRNA prep, you probably will be able to parse out differences between the RIN numbers are going to be as important as the differential gene expression. And that, you can imagine a very long transcript, for instance, that's high quality RNA where you're going to get reads all the way out across the entire transcript versus if the RNA is degraded, where the reads will essentially fall off towards the end of the transcript. In an absolute counting sense, those will look like a differential expression event when actually the gene is there, it's exactly the same, it's just that one is more degraded than the other. What we find is that those sorts of biases are not strong at all in the total RNA prep. So when I talk to people about setting up an experiment like this, I prefer to use the total RNA prep over an mRNA prep. If there's any worry about the samples being varied or having varying RIN numbers across the course of the experiment. if there's any possibility of it varying, even if they're all considered high quality in the range of 7 to 9, but some are going to be 7, some are going to be 9, then actually I might suggest using the total RNA prep as is. The RNA access prep also is less sensitive to that bias because there is no poly-A selection. It's got its own sort of bias, if you will, in that we pull out what we enrich for, but in terms of 3' or 5' bias, we don't have any of that kind of bias. So I think we're coming up to the end of our time here, and I'll try to go through as many of these questions as we can and get back to you if we can based upon this. But at this point, I'd like to thank all of you for listening and for your attention, and hope you have a good day.